Hello, I am Venmati Maran from Borneo Marine Research Institute, University Malaysia, Sabah. My title is Sea Lice Potential Pathogen of Cage Aquaculture in Asia. So, the modern technology of cage aquaculture, which was originated in Norway in the 1960s with farming of Atlantic salmon and rainbow trout, that is Salmo salor and Oncorhynchus mycis. The sea lice, Caligus elongatus and Lepiothyrus salmonis, that plague the salmon farms now were then known to occur in the wild fish and did not cause problem in the farmed fish. So this problem happened in 1970s, that the infestation of sea lice in Norway became a serious problem. Later the same problem appeared in Scotland, Ireland, France and Canada. So the sea lice problem happened in the northern hemisphere. The Atlantic salmon was introduced to southern Chile for cage aquaculture and it was spread everywhere. You can see here the parasitic crustaceans. So these are all isopods and also branchiuran and parasitic copepods. So here the important parasitic copepod is sea lice, which is causing problem in cage aquaculture. And sea lice, the wild salmon stocks in Canadian coastal waters are being severely affected by parasites from fish farms. So intense are these infestations that some populations of salmon are at risk of extinction. This is what given by the Rosenberg in 2008, published in Nature. So those sea lice or Epiphytus salmonis, Caligus elongatus and Caligus rojocracy. You can see here the sea lice, the infestation of sea lice, which is causing a big problem here. So this picture shows a typical heavy infestation of an Atlantic salmon by the sea louse Lepiothyrus salmonis, showing removal of skin over the head and female lice bearing paired egg strings on the back. So which is a big problem in culture conditions. The Asia is having a lot of the cage aquaculture production. So 90% of the global aquaculture production is coming from Asia. So the recent findings from FAO 2020, which is showing that there is a rise in global aquaculture production from 1990 to 2018, that is 527% of increase. And you can see here number of the parasitic copepods which are in different body forms. They are highly variable in body forms which are infesting the fishes. A particular copepod, that is sea lice, which is belonging to the subclass Copepoda, order Siphonostomatoida and the family Caligidae. So there are 35 genera coming under this family, which are having more than 500 species, and sea lice, which constitutes Caligus and Lepiothyrus. In northern Europe, about 30 species only have so far been discovered, but in Asia there are 250 species. You can see here the general structure of the cephalothorax of sea lice. This is Caligus because it is having the lunule. So antenna, maxillipede, these both are very important for the feeding purpose. This is the oral tube. And sternal furca, leg 1, leg 2, leg 3, these are all comprised within the cephalothorax. In general biology, you can say that the body surface, oral and branchial cavities, gills are the main region which are used for the attachment of the sea lice. They feed on the mucus, epidermal cells and blood of their hosts. They are characterized by having a mouth tube with a pair of pectinate mandibles inserted in it. The mouth tube is made up of the labium and labrum, and cubed close to the tip of the former is a pair of strigil. So the feeding time, the copepod's mouth tube is applied to the surface of the fish and tissues, including the mucus of the fish, which are scraped off by movements of the strigil. The debris is then picked up by the mandibles, which act as conveyors to move the fish into the buccal cavity. And through artificial infestation, it was proved that the sea lice can kill the host fish, and it causes a major problem in aquaculture. And there was an estimation by Costello in 2009 that The global commercial loss due to sea lice was 480 million US dollars in salmonid aquaculture. You can see here in Southeast Asian countries, these area like Vietnam, Philippines, Malaysia, all these countries are showing the infestation of caligus, so which was studied only recently. Until 2000, there was no any study on sea lice from these countries. So the main sea lice are Caligus epidermicus, orientalis, rotundigenitalis, chiostis, and epidermicus. You can see here these are the fishes are cultured in cage aquaculture, mostly the groupers and snappers. And some of the fish farms in Penang and in Tawaf, Sabah area, they were found with the infestation of sea lice in the culture of several fishes on snappers and groupers. So the parasite is Caligus rotundigenitalis, this one. And even in hybrid grouper, it was reported the infestation of Caligus rotundigenitalis. We found the prevalence as 80% and the mean intensity was 7. And the maximum number of individuals, the parasite which was found on the host, it was 21. And they were infesting mainly the head, body, and operculum region. You can see the paper, it was published in 2009. It was focused on the infestation of sea lice, that is mainly Caligus in the cultured area in Malaysia. So the main parasites were Caligus chiostis, epidermicus, Caligus longipedis, Caligus punctatus, and Caligus rotundigenitalis. It was reported from several hosts, that is Ludjanus erythropterus, E. bleacheri, and E. phascogastatus, and G. speciosus. And South Asia, mainly India, it was found with Caligus epidermicus. The cultured fishes, especially the sea bass, Latus calcarifer, Cetodus erimi, they have been reported with Caligus epidermicus. And this one is a Korean rockfish. It has happened in Korea. So this Korean rockfish was infested with number of parasites. So one host was found with several parasites, that is Lepiothyrus, Peniculus, copepods, and Calame stages of Lepiothyrus, and another genus Clavella. These are all parasitic copepods. And then monogeneans found from the gill from 3 different families we have reported. This Lepiothyrus elegans, that is a sea lice, which was found on the body surface, and the Calame was found on the fin region and pre-adult from the fin and body surface. So each stage we can collect from different region. So severe infestation was observed on the body forms. And we also studied about the life cycle of Lepiothyrus elegans. The female, it was taken and incubated. The egg sacs, they produce Nauplius 1 and Nauplius 2, and then it becomes a copepoded stage. So this is an infective stage. So this stage, which will be looking for the host, it will be attracted towards a particular host, which shows the host specificity. And the Calamus 1 and then Calamus 2, it becomes pre-adult 1 and pre-adult 2. So at this stage, in pre-adult 1 stage, we can able to differentiate the sex of the parasite, and finally it becomes an adult. So sea lice are considered as a pest. So we say that Caligus epidermicus, Caligus rotundigenitalis, Caligus chiostis, they are considered as pests in aquaculture. And already it was reported Caligus sclerotinosus infesting the red sea bream, Lepiothyrus elegans infesting the Korean rockfish as a pest in aquaculture. So this slide shows the impact on economy, that is the estimated cost of the sea lice control in salmonid production in Europe and USA. So this analysis assumed that sea lice control costs increased in proportion to production. You can see the production cost, which was US dollars 800 million, but the sea lice control cost costs 480 million US dollars. The conclusion: the sea lice is considered as a serious pathogen in cage aquaculture because the cage aquaculture has been in practice for several years in Asia, but the problem of sea lice has not yet been escalated to an alarming rate. So this is largely due to the fact that the establishment of cage aquaculture has not yet attained a large-scale operation in Southeast Asian countries, as like the farming of salmonids in cold water countries. Therefore, it is utterly important to first conduct studies on sea lice before intensive promotion of this industry is carried out. Without doubt, the transmission of sea lice occurs from the wild fish to the farmed fish. Therefore, to prevent and control the outbreak of sea lice, we need, in addition to a rich information on the sea lice fauna of Asia, a thorough knowledge of the life cycle, population dynamics, the biology, and transmission mechanisms of each species. These tasks ought to be carried out by the sea lice specialist, yet there is a limited number of specialists in Asia, which can lead in this essential basic research. Consequently, to prepare a group of specialists to work on the sea lice of Asia is considered as one of the most indispensable preparatory works to the modernization of cage aquaculture in Asia. So I like to acknowledge my grant from EMS, SDN grant. Thank you so much for your patient hearing. Thank you.